Hello, welcome to PCAP's Native Prairie Speaker Series. My name is Caitlin Mroz-Sailer, and I am the Stewardship Coordinator with Saskatchewan Prairie Conservation Action Plan, or PCAP. Today, Jolene Giacinti from Environment and Climate Change Canada will be speaking about the epidemiology of HPAIV in wild birds. Before we begin, I'd like to state that we respectfully acknowledge that we are on the traditional territories of many Indigenous nations and communities, past and present. For millennia, they have worked to protect these landscapes and the life these areas sustain. I'd like to thank these original caretakers and acknowledge the ongoing work and presence of Indigenous peoples in Canada today. Just a few notes before we get going. I'd like to mention that PCAP's Native Prairie Speaker Series is a monthly presentation about anything to do with prairie conservation or species at risk. I recently attended the Prairie Conservation Endangered Species Conference, and I was blown away by how many people approached me to say that they watched our speaker series, either live or on YouTube. So it was fabulous to hear that, and I want to thank you all for your support. I'm really excited about our upcoming webinars. Our next Native Prairie Speaker Series is April 4th about chronic wasting disease by Dr. Iga Stasia. And on May 3rd, Dr. Joe Schmutz will be talking about golden eagles. Of course, don't miss PCAP's annual Prairie's Got the Goods Week. It's a week-long series of webinars about ecological goods and services, and that's running next week, March 6th to 10th. As always, you can register for these webinars on the PCAP website. We have over 200 people registered for today's webinar, so you'll be muted for the duration of the webinar. But if you have any questions during the presentation, please feel free to type it into the questions section of the webinar dashboard at any time. If you're on a cell phone app, you can send it by chat to the organizer, and questions will be answered towards the end of the webinar. Today's webinar recording may not be publicly available afterwards, so please be sure to take good notes. Lastly, I would like to thank our Native Prairie Speaker Series presenting sponsors, Canadian Forage and Grasslands Association, Saskatchewan Cattlemen's Association, and Wildlife Habitat Canada. And in-kind support has been provided by Environment and Climate Change Canada. So without further ado, I'm pleased to introduce today's presenters. Jolene is a wildlife health specialist with the Science and Technology branch of Environment and Climate Change Canada, working on both the avian influenza and SARS-CoV-2 trials. She recently completed her PhD at the Ontario Veterinary College in Guelph, Ontario, where she also received a Doctor of Veterinary Medicine. Catherine is a wildlife health specialist and research scientist with Environment and Climate Change Canada in the Science and Technology branch since 2006. She is also an adjunct professor in the Department of Veterinary Pathology at the Western College of Veterinary Medicine, University of Saskatchewan, and has been studying the impacts of infectious diseases and environmental stressors on migratory birds for over 25 years. Catherine has been heavily involved in leading or co-leading national surveillance program for SARS-CoV-2 and AIV in wildlife across the country. So with that, I will pass it over to Jolene.
Great. Thanks very much for the introduction. Just gonna navigate to my screen here. And can I just get a verbal confirmation that you can see everything okay there?
Yes, perfect.
Great. Thanks so much. Well, thanks for the lovely introduction. I'm Jolene, and I'm really happy to be here today to discuss avian influenza, and really happy to have Catherine here as well. As you heard in her introduction, she's been studying avian influenza virus, specifically in Saskatchewan, for many years now. So she has a wealth of knowledge, and will be on during the question period to help provide all of that great historical context and wealth of knowledge that she has. Before I get started on the presentation, I did just want to clarify that I will be primarily speaking about the wild bird situation today, as at Environment and Climate Change Canada, these are the populations where we have expertise and of course a mandate for conservation and protection. There have been a number of things in the media about avian influenza in mammals, as well as some human infections that you may be aware of. And I will touch on these briefly, but I do have a resource slide at the end where there are links where you can go to get more information from the relevant agencies about those particular areas. Okay, so before speaking about the results from Canada's avian influenza surveillance program in wild birds, I want to briefly cover just some background information and what's hopefully some useful context for those who may not know that much about avian influenza. So it is a virus that primarily infects wild and domestic birds. It has been detected in mammals, including some humans, which again, you may have seen some media reports about recently, but this happens much less commonly. It is a rapidly evolving virus, which again, you may have some exposure to as well and some familiarity with just based on how COVID-19 has been progressing over the last few years. But what this means is that the genetic makeup of the virus can change at a high rate, which leads to new viruses which can behave differently. Avian influenza is also classified in a number of different ways. There are a couple that are particularly relevant to the presentation today, so I'm just going to cover them briefly. So the first is by the surface protein. So you can see from the image on the right of the slide there that in a simplistic sort of depiction like this is, there are two different structures on the surface of the virus called the H and the N proteins. And avian influenza viruses are named according to the type of H protein and the type of N protein that they have on the surface. So for example, H5N1, which is the current subtype, or for example, H12N8. So there are many combinations, and almost all known combinations of H and N proteins have been found in wild bird populations. Avian influenza viruses are also described based on how pathogenic the virus is. And pathogenic means how likely it is to cause illness or death. And when we talk about avian influenza, this is specifically relevant to or described in the context of domestic birds. So a highly pathogenic avian influenza virus is one that has a high level of likelihood of causing illness and death in domestic birds. With rare exceptions, just to go into a little bit more detail about the difference between low pathogenic and high pathogenic avian influenza, all of the avian influenza subtypes that are naturally found in wild bird populations are actually those that are low pathogenic avian influenza viruses. So these don't cause clinical signs in wild birds either. So when we're testing birds for these, we're looking to detect infection, but generally not disease as a result of these viruses. These low pathogenic viruses are commonly detected in waterfowl, so those ducks and geese, but also some gulls, shorebirds, and other species in that taxonomic group. And these low pathogenic viruses are relevant because, as I mentioned earlier, avian influenza can change. So these low pathogenic viruses can mutate to become highly pathogenic viruses. And this mutation typically occurs in domestic bird populations. So different to low pathogenic, there are actually no known natural hosts for highly pathogenic avian influenza. So wild birds are actually not considered a natural host for these viruses. So what happens is typically these mutations occur to highly pathogenic viruses in domestic poultry, and then these highly pathogenic viruses can be passed back to wild birds. With this new highly pathogenic virus, we have seen unprecedented infections and impacts to wild birds as a result of this highly pathogenic virus. So there's been a huge range of bird species that the virus has been detected in. There's also been novel or unprecedented mortality levels in a number of different wild bird species as well, globally and in Canada, with some quite dramatic mortality events. So particularly in Canada, seabirds in eastern Canada. So I have a picture there on the bottom left showing a northern gannet colony before an incursion and after an incursion. So quite dramatic die-offs in some cases. There's also been a novel number of mammals that the virus has been detected in, and I've included some photos on the bottom right over there. Of course, there's also been dramatic impacts to the poultry industry as a result of these highly pathogenic viruses beyond anything we've seen before, certainly in Canada. So I did just want to cover a little bit about what a sick bird with highly pathogenic avian influenza might look like, just so you're aware if you do happen to see any of these birds sort of on the landscape. So signs of highly pathogenic avian influenza in wild birds can include a lack of energy or movement, tremors or a lack of coordination, any swelling around the head, neck, and eyes, coughing, gasping for air, sneezing, diarrhea, but you also may encounter birds that might be dead. And I do, before I play, these images on the slide are actually videos. Hopefully they will work, but I do just want to provide a warning before I play them. They can be difficult to see. So if you do want to, step away for a moment, please take this opportunity to do so. But I think it can be useful just to get an idea of the particular signs that I'm talking about. So I'm going to play the videos now. Hopefully they'll work for you. And I'll just play that second one there. So again, I know quite difficult to watch, but it can be useful just to be aware of what sick birds infected with this virus might look like. And it is important to note that if you do encounter a dead, injured, or sick wild bird, please get in contact with your relevant provincial authority and don't handle the birds without any further instruction. So I do have a resource slide at the end of the presentation that provides more information about who to contact and where to go in that case. Okay, so talking a little bit about how the virus is moved between infected individuals. So this primarily occurs through feces and respiratory secretions. However, if there is a sick bird that's predated or a bird that's died from highly pathogenic avian influenza and scavenged, the virus can also be transmitted in this way. And this is a mechanism through which we think some of the raptor and mammal species, for example, have been infected. The virus can also survive in the environment and be transmitted in this way, such as on shoes or farm or other equipment. At a larger scale, if we think about how the virus is moved and transmitted across larger distances, there are a number of factors that are known to be important. So every year, many domestic birds are shipped around the globe, and transporting any animal or even humans moving around the world for example, can increase the chance of moving pathogens like highly pathogenic avian influenza into new areas. Although it is important to keep in mind that many countries do have important protocols in place to try and minimize the risk as it relates to avian influenza. In some countries, the areas that are most impacted with avian influenza are those with the highest poultry density. So density of industry can play a role as well. Of course, though, we know that wild birds play an important role in the maintenance and transmission of avian influenza over long distances during migration. And so to talk a little bit more about that role of wild birds, so we know that they move across large distances and in many cases in very high numbers. In North America, when we talk about wild bird migration, we typically refer to 4 continental migratory flyways that are used by a large number of birds to move between wintering and breeding areas. So there are 4 main flyways that we talk about: the Pacific, Central, Mississippi, and Atlantic. And in the middle photo there, they look like they're very defined flyways. However, in reality, what we know is that there is quite a bit of overlap that occurs between the flyways, particularly with increasing latitude. So that bottom right photo shows that with a little more detail. The timing of migration of birds does vary by species and latitude, also in relation to different habitat and climate factors. And all of this information is really useful for us when we're planning where and how to sample wild birds for avian influenza viruses. So we want to target areas where there's overlaps of birds and at a time when they're at high densities, but it can also help us understand where the virus may be most likely to move. So historically in North America, the prevalence of low pathogenic avian influenza virus is seasonal. So typically it peaks during the fall. We see a large number of birds congregate during migration, including a whole bunch of naive juveniles, so that don't have a developed immune system yet or previous exposure to avian influenza. And these infections typically decrease as the birds move south because the juvenile birds are maturing, the viral shedding is decreasing, and the birds tend to occur at lower densities in many cases. There are also several transoceanic flyways. So on the global scale, there are 9 major flyways that are described, several of which are transoceanic, connecting Canada with many other countries around the world. And a couple of these flyways have been implicated as possible routes of incursion for the current and historic highly pathogenic avian influenza viruses. I want to review a brief global timeline just of the highly pathogenic subtype that's relevant to the current outbreak. So you'll note that throughout the presentation, I'm going to be talking about H5, highly pathogenic avian influenza, and this is related again to that surface H protein. So we call it subtype H5, and that's the particular virus that's relevant to the outbreak today. So I just wanted to draw your attention to that. So the lineage of highly pathogenic avian influenza H5 virus that's relevant emerged in poultry in Southeast Asia more than 25 years ago. During the first decade of circulation, the virus diversified into multiple genetic clades, and then for the first time in 2005, it spread to Russia, Western Europe, Africa, and the Middle East. And for the first time, it caused high mortality in some wild bird and poultry facilities. With this increasing activity globally, Canada actually started its avian influenza surveillance program in wild birds, and this program has remained since that time, but it's waxed and waned in terms of the level of surveillance needed just based on what was happening on the international scale. In 2014 and 15, there was a new intercontinental spread of H5 highly pathogenic virus. And so we had the first detections in North America, and it was detected in BC and Ontario, but was primarily in poultry and was stamped out. So it didn't persist. Between 2018 and 2020, there were again increasing reports of H5 highly pathogenic viruses in wild birds and poultry in Europe. And in light of this, Canada again began to ramp up the surveillance program in wild birds in order for us to be able to detect any possible new incursions in Canada. In December 2021, there was a die-off of domestic birds on an exhibition farm in St. John's, Newfoundland, and shortly after, sick and dead wild birds that were confirmed to be highly pathogenic H5N1 positive. And this was the first report of this virus in the Americas since 2015. An analysis of the sequence of this virus determined that it was closely related to that which was circulating in Eurasia, implicating again those transoceanic flyways as a possible and likely route of introduction. So since these initial incursions, the virus has been detected in every province and territory, the details of which will be the focus of the large majority of the rest of the presentation. And of course, over 2022, we had widespread impacts to the poultry industry, but also in our wild birds in Canada and the US as well, and now even into South America as birds have migrated south. So I wanted to draw your attention to a couple public-facing dashboards which do provide up-to-date information on confirmed detections in both Canada and the US. I recognize you probably can't click the links on the slide, but we'll make sure to circulate those after the webinar. So this is a way you can stay up to date on any confirmed detections of the virus in Canada and the US, in wild birds and in poultry. Okay, so now I'm going to transition to talking about Canada's interagency surveillance program in wild birds, why and how it's carried out, and then discuss some of the results that we've had since the outbreak began. I do want to start, of course, by acknowledging the huge number of collaborating partners that we have that are involved in wild bird surveillance, from our federal governmental partners to our provincial, territorial, and Indigenous partners. We have partners in academia, and even partners in the US and globally as well. So this file really is a One Health file where we're engaged with many, many different partners who help us to deliver this surveillance program. So this surveillance is conducted to tell us where, when, and in what species the virus is present, what the impacts of the virus might be, and for us, this is specifically related to wild birds. So the Canadian Food Inspection Agency and provincial ministries and public health agencies focus on the domestic animal and human health side of things respectively. But again, we do all come together and work together on this One Health issue, but primarily at ECCC, our surveillance tells us, and we look for what the impacts are on wild birds. We also want to know how the virus might be changing over time. And all of this information is important to know, of course, so we can protect our wildlife in Canada. It's also important so we can generate information that's needed for agencies and stakeholders to help inform their decisions that need to be made regarding biosecurity and biosafety protocols in the poultry industry. Also to generate information to help folks in public health make the decisions that they need to make. We also have international reporting obligations, so we are required to carry out surveillance and report our results to the World Organization for Animal Health, as well as it helps us to have situational awareness, so to tell us what's going on with avian influenza in our wild birds, and also our surveillance is set up to help us try and detect any new incursions of foreign highly pathogenic viruses as early as possible. And the way that we carry out this surveillance is through a couple different mechanisms. So we have our morbidity and mortality surveillance. And so these are sick and dead birds that have been detected and submitted often by members of the public and then tested for avian influenza virus. And this tells us about what birds are getting infected and dying from this virus. We also have our live and hunter harvest surveillance. And these are the sampling of apparently healthy birds. And usually this is done alongside banding programs or research programs that are ongoing already. And so we add in avian influenza sampling. We also work with harvesters to be able to sample those birds as well. And the reason these are lumped together is that these animals are these birds in particular are those that we're looking for evidence of infection, but that don't die. So these are birds that are most likely to be able to carry the virus around. There are also other sampling activities that are undertaken. So for example, we do have areas where we sample the environment, so the water and the soil. I'm not going to be discussing these results today, but that is an important component to think about how and where the virus may be persisting in the environment. And then I have mortality events. So it's also important for us to count all of the birds that might be involved in a die-off event. So if there's a large number of birds, we typically don't need to test them all. So we test enough to know that that die-off was related to avian influenza, but we prioritize our resources and don't need to test every single bird in that group. But it is important that we do track all of those birds so that we can use that for our population level impact assessments. So a little bit separate from our surveillance program, but still a really important part of some of the efforts that are ongoing. So when we have a bird, how do we sample it for avian influenza virus? Well, we collect two different swabs. One is from the cloaca and one is from the mouth. So these are the two ways that the virus tends to shed in these individuals. So we collect two different swabs and then we put them together in the same vial and send them to the lab for testing. The background picture here, I believe, is from Last Mountain Lake in Saskatchewan, where Catherine's field program has been sampling live wild birds for many years now. So we have a great historical data set from that area, and Catherine can maybe answer any questions about that during the question section. I did just want to highlight that when we collect these swabs and send them to the lab, we get information back about viral shedding. So we're testing birds for the presence of the actual virus itself. In apparently healthy birds, there's a pretty short window where we're able to pick up these infections. So this is important to keep in mind when we're interpreting our results because it tells us about the situation during that specific point in time. Another way we can test birds, though, is by collecting blood. And this lets us test for antibodies, which can tell us more about previous exposure to the virus, where birds have developed an immune response to that. We are working to increase our blood sampling in Canadian wild birds so we can get a more clear picture of the level of exposure, but I do have some results to share with you, particularly from Catherine's surveillance in Saskatchewan. Okay, so I'm going to dive into some results finally from our surveillance. I'm going to start with the results from our sick and dead bird surveillance since the start of the outbreak. So in the data set, there's record of over 6,500 sick and dead wild birds that have been submitted and tested since November 2021. A small percentage of these, which you see in the blue, the far right color there, are samples that have been collected recently, and we're still waiting for results on those. But for the other categories, it's important to note that there's a number of different tests that are carried out for avian influenza. So first, we're testing for what subtype, so what H type and what N type are present on the virus. But then we also test for whether it's a highly pathogenic or low pathogenic viruses. So there's a number of different tests that are carried out. But for the purposes of this slide, the way that we can think about it is the red and orange categories are those samples that are either confirmed or suspected to be highly pathogenic avian influenza. So in the total data set, we have roughly 25% of sick and dead birds that have been either confirmed or suspected to be positive. The yellow category are samples that are likely to be low pathogenic avian influenza viruses. And there have been relatively few detections in sick and dead birds, whereas historically, without this highly pathogenic virus, we would have expected to see that more commonly. And then of course, the gray, the largest category, are those birds that were found to be negative for avian influenza. And the colors of these results will remain the same throughout the presentation, just for your awareness. This is the same visual, but for Saskatchewan only. So over 800 birds were tested for avian influenza since November 2021. Again, these are sick and dead birds. So the proportions look roughly similar to the national picture, a small proportion with results pending, a combined total of roughly 30% that were either confirmed or suspected to be highly pathogenic avian influenza, and then relatively few low pathogenic detections. This slide shows where sick and dead bird submissions have been occurring, as well as a breakdown of results that are in the table there. So the size of the pie chart reflects the relative number of submissions, which is also shown in the table. These proportions are crude, so they do represent numbers over time and across species. So it's important to keep that in mind. But some high-level takeaways from this slide are that submissions were processed across the country, so all provinces and territories. Avian influenza detections were largely highly pathogenic in sick and dead birds, and generally a lower proportion of birds in Canada's north were found to be highly pathogenic avian influenza positive. However, a number of birds were also found avian influenza negative, and this can reflect a couple of different things. So there's a wide distribution of species that are submitted, and there's different susceptibilities among species to this virus. So it could reflect just the types of species that are submitted through these surveillance programs. But also it is a good reminder that there are other causes of morbidity and mortality occurring in wild birds that are still happening in the background of the outbreak. So at ECCC, when we think about wild bird health, we always try and bring all of these different components together and think about the cumulative impacts of all of these different pieces and how they work together to impact the health of our birds. So this next slide displays the number of submissions and highly pathogenic avian influenza virus detections over time in sick and dead birds. So there was a ramp up of submissions between January and February. There appeared to be a peak in both submissions and detections in April and May. Submissions then decreased, but if you look at the proportion of the red and orange parts of the graph there, you can see that the proportions increase again in the fall, which is what we would expect based off of what we know about the seasonality of low pathogenic avian influenza, which we discussed earlier. So those birds congregating at high densities, the increase in juveniles during that time. But again, we know that these numbers only reflect those birds that were submitted and tested. So we also have to keep in mind whether submission patterns were changing or there was different priorities for what birds were tested. So there's always a lot of context to keep in mind when we're interpreting these results. If we take a look at the same figure, but for Saskatchewan, we see a similar peak in submissions and detections in April and May. and a possible second wave in the fall, again with that increased proportion there, even though submissions had decreased quite a bit at that time. There do continue to be detections both in Saskatchewan and across the country, but in much lower numbers. And so that's good news, but we do know that there are continued detections and spread in the southern wintering grounds. And so we are anticipating a resurgence of cases later in the spring, coinciding with the birds moving north again. If we take a look at the species breakdown of these cases, so there were 218 different species that were screened through our sick and dead bird surveillance, and 86 of these were confirmed or suspected positive for highly pathogenic avian influenza, which is consistent with that increase in host range in wild bird species that I mentioned earlier. If we take a look at a tree map, this shows the relative number of different groups of species that we've had detections in. So we see the bulk of the detections have been in waterfowl, so that light blue color, so these are our ducks and our geese. In raptors, so that white color, we have our hawks, our eagles, our owls. Passerines in that sort of lighter green, these are our songbirds and our corvids. Largely in passerines, it's corvids. And then we have what are called charadriforms. This is the taxonomic group, and it contains quite a diverse range of species, but things like gulls, shorebirds, murres, and a number of others. And then we have the yellow category, which is our seabirds. If we look at this in a little bit more detail, this only includes sort of the 5 most numerous species groups, but we can see again, so that green color, corvids make up the large majority of passerine detections. Gulls, if we look at that darker blue color, make up the majority of that group of birds called charadriforms. Multiple raptors were represented, and then geese make up the majority of waterfowl detections, and dabbling ducks a much lower amount. But again, it is important to keep in mind that many birds that died were not submitted and tested, so this doesn't reflect the total number of birds that are expected to have died as a result of highly pathogenic avian influenza. So for example, we see northern gannets in that yellow color, and we know that the scale of mortality in this species is quite a bit beyond what's shown in this figure here. This is a short time series showing the highly pathogenic avian influenza detections in sick and dead birds over time. So I'm going to play the video and talk through it a little bit. So we start with the index case in gulls of the Atlantic region, quickly moving to detections in raptors and waterfowl in the same region. Then we move to BC and southern Ontario where we see detections. And then as birds continue to come in from the south, we see that the prairies start to light up. And then detections move north with wild bird movement. I'm going to play the video again, but I did just want to note an interesting pattern. So after the index case in the Atlantic region, the first detections in other new areas across southern Canada were typically in either sick and dead waterfowl, so the blue dots, or raptors, the white dots, and then followed generally by corvids, which are the green dots there, and then filled in with other species. And this pattern really is replicated across new areas detected in southern Canada. As detections move north, the first detections in many northern regions were charadriforms and specifically gulls. So I'm going to play the video again for you and just take a bit of a water break so you can watch it. And just another interesting note that these trends in the order of detection of species make sense if we think about infected waterfowl being detected first. They do occur in high numbers in areas where lots of people tend to be. And then a likely route of exposure for some of these other species is through scavenging or predation of these infected waterfowl. So if they're displaying abnormal behavior, they may be more visible to predatory or scavenging birds, making them easier prey. So, you know, our species like raptors and even some of the mammals, this is considered to be a likely route of exposure for those species. If we bring some of the information about sick and dead birds together with some of the infected premise information, so these are those domestic poultry facilities that had reported outbreaks, we can see some interesting trends. So this slide shows Saskatchewan specific data. The black bars in this first table at the top are the number of domestic facilities with confirmed infections over time, and those are on the right axis. And this was last updated in November, so there may be additional information that needs to be added after that point, but this is just to give a general idea of some trends. The red line in this graph shows the number of sick and dead birds that were confirmed to be highly pathogenic avian influenza positive through the surveillance we've just been discussing. So we see that peaks in sick and dead birds correspond with peaks in domestic poultry infections. The arrows here indicate the first sick and dead birds that bird that was detected in Saskatchewan. So this was a snow goose. The first arrow is the date it was collected, and the second arrow is the date that the results were first available. So what this tells us is that we were actually able to detect the presence of the virus in wild birds through surveillance before the first detections in domestic birds, which is one component of that early warning piece that I mentioned before. We can see these locations, the location of these detections on a map relative to the rough location of the domestic facilities. So the domestic facilities are in black and the first wild birds detected are in red. And it is important to note that the location information here is approximate for privacy reasons. So the location information used is only that which is publicly available. So I did just want to highlight that, but it does show you the rough geographic location relative to the wild bird detections. And then the final visual shows the same domestic poultry information as the first graph, but the lines now instead represent species frequency data that we obtained through eBird for some key species in the province. So I use the short forms here, but this means blue-winged teal, Canada goose, green-winged teal, mallard, and snow goose. So you can see that migrations of key species correlate well with the peaks of highly pathogenic avian influenza detected in wild birds, but also in domestic poultry facilities. And then I do have the same type of visuals, but this is for Ontario instead. So you can see that the trends are similar in different areas of the country. I'm going to move to some of our live and hunter harvest results now. So the visuals will follow a similar order, but instead we're looking at apparently healthy birds. And so remember, what this tells us is about those birds that are exposed but surviving infection. So in total, we have over 11,000 samples from apparently healthy live birds tested since November 2021. Again, a small number have results pending, that far right category in blue. But if we look at orange and red, there's a combined total of 5.5% of live and hunter harvest samples that have been suspected or confirmed to be highly pathogenic positive. Only a very small number have actually been confirmed in live birds. There are more low-path avian influenza detections in these apparently healthy birds, and that's reflected in that yellow category there. And then again, we have our avian influenza negative, which is almost 84% of the samples. This is the same visual, but for Saskatchewan. So again, very similar trends. A higher proportion of that yellow category, so those low pathogenic avian influenza viruses. We actually don't have any confirmed highly pathogenic viruses in Saskatchewan at this time, but 8.4% of samples are in that suspect category. So they're still undergoing that confirmatory testing, which can be a number of different steps and can take quite a bit of time. In Saskatchewan, these suspect detections were largely geese. If we move to the map, we can see that low pathogenic avian influenzas were detected in a higher proportion across the country as compared to the sick and dead birds. And again, it's not unexpected for us to see these low pathogenic viruses in our wild birds. Whereas the proportion of those suspect or confirmed highly pathogenic positive samples does vary quite a bit at the provincial and territorial level, ranging from 0 to 36%. So some of the higher percentages, so that 37% was in Nova Scotia, and was largely driven by mallards, and teal and black duck at various times of year. And we are awaiting sequencing on these samples to confirm again that the highly pathogenic nature of those. If we look at sampling over time, we can see that the majority were collected in late summer and fall, and this corresponds to when most banding programs and research programs occur in our migratory birds. The peak of detections of low pathogenic avian influenza, so that yellow piece of the visual there, also occurred in the fall, which again is consistent with that seasonality that we talked about before. If we look at species for our live and hunter harvest birds, the sampling is largely done in waterfowl, gulls, and shorebirds. And overall, there were 68 different species that were sampled, 22 of which were suspect or confirmed highly pathogenic positive. Again, if we look at that tree map, we see the trends are largely driven by waterfowl. And if we look a little more in detail, we see that the detections were largely in healthy dabbling ducks, some geese, and some Mroz. And this is not an unexpected distribution based on what we know about low pathogenic avian influenzas in live birds in Canada in the past. Dabbling ducks are known to be an important host for this virus, and we're still learning about whether they will play a similar role for highly pathogenic avian influenzas moving forward. So this is just a quick summary slide because I know you folks might be particularly interested in some of the species that were detected in Saskatchewan. So in sick and dead birds, they were primarily passerines, raptors, and waterfowl, so specifically crows and magpies, eagles, hawks, owls, falcons, vultures, a number of different raptors. The waterfowl were primarily geese and a range of geese species. And then through live and hunter harvest, the suspect detections at this time are in snow geese and northern pintail, largely the snow geese though. So I'm going to talk briefly about a couple species-specific results here. And so first I'm going to start with blue-winged teal. And this is a big component of Catherine's testing program that she's been running for many years. These species are particularly important because they're long-distance migrants and they've historically been important in understanding the dynamics of low pathogenic avian influenza viruses. Large mortality events generally were not reported in this species in Canada, so that's just important context to keep in mind. So 397 apparently healthy blue-winged teal were sampled at Last Mountain and Porter Lakes in Saskatchewan in August 2022, and they were tested both for the presence of the virus and the presence of antibodies for whether they had developed immunity against exposure. Most of the birds sampled were juvenile, 23% were adult, and sort of some of the big takeaways from this sort of preliminary analysis is that no highly pathogenic avian influenza virus was detected. And this is actually similar across the country for this species. We didn't detect virus in too many of these birds. All the virus detections that we did find were low pathogenic, and the majority of these were in juveniles. So this sort of coincides with what we know about this time of year in juveniles. So they appear to be the most vulnerable. They're developing their immune system. They haven't had that past exposure to avian influenzas. So not unexpected there. If we look at exposure, so our antibody testing and the blood samples, the majority of juveniles did not have evidence of exposure to avian influenza viruses yet, or highly pathogenic avian influenza viruses. So there's still quite a high proportion of these birds that are naive to avian influenza. Whereas if we look at the adult birds, quite a higher number had exposure to avian influenza viruses, which again is to be expected, but very few of these were to highly pathogenic avian influenza. So there are different tests to differentiate what type of exposure these birds had. And so again, what this tells us is that there's still quite a high proportion of adult birds, at least in this area, this population, that are naive to the highly pathogenic avian influenza virus. If we take a look at geese on the other hand, so we have snow geese on the left here and Canada geese on the right. The bottom graphs show those geese that were detected sick and dead with the virus. So quite a number more geese detected to have died as a result of the virus compared to the teal that we just talked about. And this occurred sort of throughout the year and across the country. If we look at the healthy birds, which are the two top visuals, we actually found very few detections in healthy Canada geese, some suspect detections in the snow geese. But what we're really sort of interested in and wondering about is what the level of exposure are in these geese. So have they been exposed? Have they developed that immunity? And we are waiting on some test results of blood samples that we do have from these species. But the good news is that there's some early results from the US in snow geese in particular that have shown that quite a high proportion of these birds have been exposed and so may have some level of immunity against this virus. And that might mean that mortality events seen moving forward may not be quite as significant as they were last year. But again, these are some early results, and so we're still trying to learn more and understand, what to anticipate moving forward. So that's the end of the results section. I did just have a couple notes about sort of where we go from here and what Environment and Climate Change Canada is doing moving forward. So given the continued detections and spread of highly pathogenic avian influenza in the southern wintering grounds, we are anticipating a resurgence of cases in the spring of 2023, coinciding with, again, that northward migration. And so in light of this, Environment and Climate Change Canada continues to work with all of our partners to deliver our surveillance. So we'll be continuing sick and dead bird surveillance, live and harvested bird surveillance. We also continue to monitor our wild bird populations where that did experience those mortality events as a result of the virus. So we're assessing those for any potential population level impacts. We do, of course, are thinking about whether there are any implications for our species at risk in Canada. And we do have a number of targeted research projects that are on the go to increase our understanding. And then, of course, we work with all of our partners to make sure we're communicating all of this information out via our websites, dashboards, and other social media and through presentations like this. So this is that resource slide I mentioned before. A number of great guidance materials are available online. So of course there's general and public health guidance and hunter harvest guidance. So there's the Government of Saskatchewan and the Government of Canada websites here. If you're looking for those updated reports on the current situation, I've provided the link for the Canadian dashboard, the US dashboard, and then international information there. To learn about detections in domestic poultry, the Canadian Food Inspection Agency does have this information available on their website, as well as the Government of Saskatchewan has additional information as well. And then if you do encounter any sick and dead birds in Saskatchewan, these get reported to the Canadian Wildlife Health Cooperative, and I have provided the link there for you. And I'm going to end there, just again on this slide, recognizing all of our many partners and collaborators on the file. Thanks very much.
Thank you. That was a really awesome presentation. We've had a lot of questions come in, so I'll try to give them to you rapid fire, and between Jolene and Catherine, feel free, whoever think is best to answer them. So the first one is, do you think that raptors will display similar behavior actions as depicted in the videos that you showed?
Sorry, I just missed the first part of that question. You cut out a little bit there.
Will raptors display the same behavior as shown in the videos?
Yeah, that's a great question. As far as I understand, the raptors that are not being found dead, the ones that are being found with clinical signs, are displaying neurological signs as well, so behaving abnormally. I don't know, Catherine, if you have anything additional to add to that.
Nope, you got it. That's what it is. So oftentimes they're just found dead, or if they're ill, it's usually neurological signs, abnormal behavior, not afraid of humans.
Thanks for that answer. Great horned owls are waterfowl predators. Are they more susceptible to the virus?
Yeah, so that's another great question. So susceptibility to the virus is a little bit different from likelihood of exposure to the virus. So we know that raptors in general seem to be quite susceptible to highly pathogenic avian influenza virus. And we have seen a number of birds found dead as a result of this virus. But in terms of likelihood of being exposed, certainly species that do scavenge on birds that are likely to have the virus would have an increased risk as well.
Okay. And then on one of your slides about confirmed/suspected HPA positive, you had a other raptor section, and one of our attendees is wondering if falcons fall under hawks or other raptors.
Other raptors.
Okay.
Sometimes it's a little bit hard to know how to categorize things, but yes, that's a good question. They do fall under other raptors.
Absolutely. Do you work with research teams that are already collecting blood samples for their own research? For example, have a CWS or provincial permit? I'll leave it there.
Yeah, that's a great question. So we have a large sort of network of research programs that we do have close relationships with across the country. We're of course always interested to collaborate if you are aware of other opportunities. But yeah, we work with many to collaborate on avian influenza surveillance research.
Perfect. Is it possible to test historically collected samples for retroactive review?
Yeah, so that's another great option that we have. So it depends on the sample a little bit and how it's been stored, but certainly there are opportunities for archived samples to be tested for avian influenza, and we have done quite a bit of that. I think Catherine has done with some of her work to look at historical avian influenza. But again, this virus is quite new to the area, so the historical samples we wouldn't anticipate to be testing positive for these highly pathogenic viruses.
Okay, perfect. And then would you be able to go back one slide with the links?
Yes, of course. And I can always make sure to circulate those as well, because I don't think you have the ability to click on them through the webinar. So some of them are long, but we'll make sure to share that.
Yeah, and people can do a screenshot as well, but I do see there's a lot of numbers and letters. They're not easy links. So yeah, that would be great. When a wild hen duck is able to shed the AIV, are there maternal antibodies given to the young when they hatch?
Yeah, that's a great question and something we've been looking to research a little bit more. We do know that there's transmission of immunity in that way, but learning a little bit more about how long it lasts and how likely it might be to help protect against this current highly pathogenic virus. But Catherine may have a little more insight into that as well.
Yeah, I was just going to say that, so if there is a transmission of antibodies, via the yolk to chicks as they're developing in the egg, they likely would not last very long. Typically in mammals, those tend to last about 2 weeks, depending on the virus. So as that wanes, then they become more susceptible when they're exposed. And there is something that we're also interested in is how much cross protection is there. So if birds have been previously exposed to low pathogenic strains of influenza, which cause no clinical signs, is there some protection if they're subsequently exposed to this H5 highly pathogenic influenza? And maybe that is why we find quite a few dabbling ducks that are apparently healthy and are shedding the virus. So that's something that would be super interesting to examine.
For sure, for sure. There's a few people that are wondering about where they can report dead birds in Alberta. If either of you don't know that, then I can ask the audience as well. So there's people here who might write in the answer.
I do have that information. I'm more than happy to share it. I don't have it at the tip of my fingertips right now, but I do. It's the Alberta Environment and Parks Office, and I do have a phone number that I can share maybe when I share the resources as well with you.
Sure, yeah, that would be great. Perfect, thank you. Do hunting Do hunter-harvested birds pose any issues for people eating the birds?
Yeah, so that's a great question, and something that we've worked closely with public health colleagues to develop guidance and recommendations for. So I will direct you to the resources online that are available to sort of give you a lot more information. But just in general, what I can say right now is that while we've detected it in migratory birds, at this time, if they're properly sort of cooked and consumed, the risk is considered to be minimal. But again, I would refer you to those sort of more detailed guidelines that are available online.
Perfect. And then what about for hunting dogs? Is there a risk of exposure for hunting dogs that retrieve a bird in its mouth?
Yeah, so that's certainly something to consider. And of course, we do recommend, where possible, not letting your dog interact with any birds that are appearing to be sick or dead, keeping them on a leash where that might be a risk. And then of course, talking to your veterinarian about the specific risk for your dog and your lifestyle. But certainly we would want to avoid that interaction with any infected bird.
That is good to know. What can you tell us about the survival of infected birds?
Yeah, Catherine, maybe I'll give that one to you.
That is a really good question, and that will most certainly depend on the actual species. So there are some species that we can see right now are highly susceptible, high rate of mortality, like those raptors and corvids, so that if they're infected. But when it comes to those carrier species or the natural reservoirs for low pathogenic avian influenza viruses, they seem to be a bit more resistant to the virus. And so we don't know what the survival rate is for, say, a mallard that has been infected and survived. Does it have long-term impacts as well? So the virus may not kill them outright, but perhaps it affects their speed of migration, or quality of habitats they select, or they die en route, through migration. So those are things that we hope to be able to tease out now that it seems to be here, and we can do that in a number of ways. So if we continue doing the surveillance program like we continue that we plan to do, we're also banding birds. So capturing live birds, banding them, and there's reporting system that if you hunt a bird that has a band on it, you can report the band, and we collect that data as well and can do some models to look at infection in relation to survival, or infection in relation to probability of being hunted. And over time, over years of collecting data, we'll have more and more accurate estimates on survival, but at this point we don't know. There could be experimental infections that happen in captivity that could help us answer that question as well, and I believe the CFIA has been interested in doing some of those to look at vaccination and then infection in waterfowl, or previous exposure to low pass strains and then subsequent infection, just to understand how that affects waterfowl. Does it, how quickly do they die, or do they die, or how long do they shed the virus? Is it 3 to 10 days, or so those are lots of questions that we're still trying to pinpoint, and it may change with the different strains of viruses we are finding. So this avian influenza viruses evolve very rapidly, and they can mix with the viruses that we have here in Canada, North American strains, to create new, what we call reassortants of viruses. So do they have differences in how well they are at affecting birds or killing birds? So it'll depend on species, it'll depend on the viral reassortments. And complicated question.
It is very complicated. There's a few people asking, so. Do you know how long the virus can survive in the environment, or specifically in a wetland?
Jolene, do you want to start that one?
I'll start it. Yeah, it's another great question that doesn't have an easy answer. It does depend on a number of different environmental conditions. So temperature, salinity, humidity, the load of the virus, also impacts the risk of being exposed in the environment. So there are a number of different factors that are relevant. So in some cases, we would not expect the virus to survive very long. The conditions were not favorable, but we do know that if the conditions are favorable, that the virus can survive for potentially long periods of time. I don't have a specific number in my head, Catherine may, but a lot of what we know, at least about survival of the virus in the environment, is actually those low pathogenic viruses. So we have some data about high pathogenic viruses based on studies in the laboratory, but how those might translate to sort of the environment in Canada, I think, is something we're still learning about and trying to understand.
Thanks for that answer.
Yeah.
When you share the
you got that, Jolene. Another thing to add is some folks have found that low pathogenic strains of avian influenza can overwinter. So will that be the case with highly pathogenic strains of influenza? We don't know yet. So hopefully we'll be able to do some environmental sampling this spring to see.
Absolutely. And when you send the resources, could you also include a resource for Manitoba, what people can do there?
Of course, I can include the resources for all the provinces and territories, so we can just cover all the places. Yeah, it's so great to hear that folks are interested in reporting sick and dead birds. It is a really important component of our surveillance program. So it's always great to hear that folks are interested in helping us out with this work.
Absolutely. Do you have any best practices for people with bird feeders?
Yeah, that's a great question. So I can include, I believe one of those resources does have our recommendations for backyard feeders. The recommendation currently is that feeding sort of typical backyard birds, songbirds, is low risk, but to just make sure you're regularly cleaning your feeders in the appropriate ways. And so I can direct you to the resources for that have a bit more information.
Okay, that'd be perfect. I think that's all the time that we have today. We've kind of taken up more than an hour, but I want to encourage people who didn't have a question just to kind of reach out to you directly, Jolene or Catherine. You're a great Saskatchewan resource here. So I know there's been lots more questions coming in than we could handle today, but I want to thank you both for coming and sharing so much information. This was really, really informative, and I know really well received by our attendees. Lots of comments already about how much people have enjoyed this presentation and how informative it was. Thank you.
That's a real pleasure. Thank you.
Yeah, real pleasure. Thanks for the invitation. And I have just gone back to the first slide that has my email address there. So please do feel free to reach out if you do have additional questions, and we'll be sure to share those resources.
Okay, perfect. And I just want to reiterate a comment that just came in. Thanks for sharing all this information and keep up the good work. Oh, that was very nice.
Excellent.
So to all of our attendees, thanks for catching today's webinar. Hopefully I see you online next week for Prairie's Got the Goods Week. We have 7 webinars going on, so definitely take in some of those if you can. And our next Native Prairie speaker series will be in April about chronic wasting disease. That's it for now. And when you leave today's presentation, if you don't mind filling out a quick 1-minute survey, we really appreciate it. And it gives us ideas what to cover for the future, as well as we can report back to our funders. So with that, thank you very much, everyone, and have a great rest of your day.